a critical role in the maintenance of mitochondrial DNA. Dysfunction in this particular signaling pathway has been associated with both neurodegeneration and cancer. When it comes to retrograde signaling, it is the crosstalk from mitochondria to nucleus, and it is already well known that mitochondrial ROS has been linked to regulation of gene expression in the nucleus. Recent years, there are a couple of papers which have shown that mitochondria can indeed encode some of the non-coding RNAs which are listed here, like long non-coding RNA, circular RNA, and also microRNA. So one of my PhD students, Ravi, he performed some in silico bioinformatic analysis to predict that mitochondrial genome encodes 13 microRNAs, which are shown here. Further, what he did is he did a small RNA sequencing analysis to identify the abundance of this predicted microRNA in different breast cancer subtypes. What is shown in the right-hand image is a circular diagram of the mitochondrial genome and the abundance of the predicted microRNAs, which are shown in MDMA468, which is a triple negative breast cancer, MCF7 is a ERPA positive breast cancer, and MCF10A is a breast epithelial cells. Subsequently, we also shown that these microRNAs which are predicted in the mitochondria also interact with the RISC complex protein. Now, RISC complex proteins are RNA indesilencing complex proteins, and one of the protein is argonaute 2. This argonaute 2 protein is present in the mitochondria, and this further interacts with the microRNAs which are predicted in the mitochondria, both in the cytoplasm and also in the mitochondria. We wanted to know the localization of this microRNA which is predicted in the mitochondria. What I am showing you is the localization of this microRNA in both its cytoplasm and mitochondria. The first image is staining for mitochondria using microtracker red, and then we used the probes which are tagged with biotin, and using a fluorescent molecule, we could able to show that this particular microRNA, which is predicted by us in the mitochondria, indeed overlay with the microtracker red staining, suggesting that they are present in the cytoplasm and in the mitochondrial region. Subsequently, we looked at the expression pattern of this microRNA, which are coming from mitochondria in different breast cancer subtypes. And what I'm showing you here is the microRNAs, the location of the microRNAs in the mitochondrial genome, and their expression pattern in different breast cancer subtypes. What we understand is this microRNA have differential expression in different subtypes of breast cancer, suggesting that they might play some functional role in the breast cancer. We also wanted to know whether these microRNAs are also expressed in the patient sample. So what we did is we did a clinical study with the breast cancer patients who are subdivided into different subtypes, luminal A, basal, and HER2 positive breast cancer. And we did a paired sample analysis where in the normal tissue you can see clearly there's a decreased expression of the microRNA, and then the paired tumor tissue from the same patient you can see there's an increased expression of the microRNA, and this pattern is pretty evident in most of the subtypes in the breast cancer. This gave us an idea probably these microRNAs are specifically expressed in the cancer tissue when compared to the normal counterpart. So this data is already published in Journal of Cell Science in 2022. Subsequently, the other PhD student in our lab, Amulya, she did further analysis to understand how this microRNA expression pattern is regulated in different breast cancer subtypes, what are the target genes, and what is the functional role of this microRNA. During the in silico analysis, what she identified is out of 13 microRNAs which are produced by mitochondria, 11 of this microRNA targets a single gene which is ZEB1. Now ZEB1 is a transcription factor and it is primarily involved in epithelial to mesenchymal transition in cancer cells. So this is a classical EFT mechanism where you can see the epithelial cells get transformed into a mesenchymal cell phenotype, and this EFT process is governed by a set of core EFT transcription factor ZEB1, ZEB2, snail slug, and twist. So ZEB1 is one of the core EFT transcription factor. Expression of ZEB1 promote this transformation of epithelial cancer cells into mesenchymal cancer cells. So when we look at the microRNA expression, which is targeting ZEB1, you can clearly see compared to the MCF10A, which is the normal breast epithelial cells, microRNA3 expression is high in MDMB468, which is more of a basal-like epithelial breast and triple-negative breast cancer, and it is downregulated in MDMB231, which is mesenchymal triple-negative breast cancer cell line. So we looked at the ZEB1 expression in different breast cancer subtypes, and what I am showing you here is the expression of ZEB1 in MDMB468, where the mitochondrial, one of the target, is upregulated. You can clearly see it is downregulated. So ZEB1 is downregulated in most of the breast cancer cells, barring some of the breast cancer cell line which have mesenchymal phenotype. MDMB231 is one of such cell line where ZEB1 expression is pretty high. So we wanted to validate this finding by developing different cell line models. So what we did is we used mimics which basically upregulate the expression of mitochondrial 3. Since 231 it is downregulated, so we use this mitochondrial mimic transfected into 231 cells to upregulate this microRNA expression to see how it regulates ZEB1 in this MDMB231 where ZEB1 is upregulated. We also use an inhibitor which basically blocks the function of mitochondrial 3, and we use that again MDMB468 where this microRNA expression is upregulated. And to make a stable loss of function cell line, we use mitochondrial sponge. So in the sponge cells, what it does is it basically blocks the binding of the microRNA to its target gene, and it's a stable cell line. So how the sponge cells work? So basically you have a viral plasmid which continuously produces this antisense oligo, and this antisense oligo goes and binds to the microRNA binding site. We call it a C sequence, and by blocking the C sequence, there is no change in the expression of the microRNA, but it basically prevents the binding of the microRNA to the target gene. So essentially, you are rescuing the expression of the target gene in the cancer cell. So in our study, it is the ZEB1 which we wanted to study. So here is the data for the luciferase acid. So this is a simple biochemical assay to confirm whether this microRNA indeed binds to ZEB1. And by performing luciferase assay, we showed that yes, it is targeting ZEB1. When we created the sponge cell, we wanted to know whether by making the sponge, we can rescue ZEB1 expression in the sponge cell. So in the MDMB468 where ZEB1 is downregulated, we could able to see that ZEB1 high expression in the sponge cells when compared to control cells. Subsequently, we did immunofluorescence analysis, and you can clearly see by comparing with the control cells where ZEB1 is not localized to nucleus, in the sponge cells you can clearly see ZEB1 is now localized to nucleus. Now what is the function of ZEB1?